The overall goal of this DNA assembly protocol is to enable the repeatable, scalable, automated, high-throughput production of DNA devices.
This work addresses the need for a robust and user-friendly software workflow that can generate pipetting instructions for a liquid handler based on high-level DNA device designs.
The main advantage of this technique is that the automated reaction preparation is repeatable and scalable, and requires as little as 4% of hands-on time compared to manual preparation.
Visual demonstration of this method is critical because the automation steps can be difficult to learn and are a combination of experimental and computational methods.
A software tool that can automate script generation was developed for this study. Using any web browser, navigate to mocloassembly.com and upload GenBank files for all DNA parts that will be included in the combinatorial DNA device design. Once all the files have been uploaded, select the desired DNA parts. Collections of parts as well as individual parts can be selected. Drag the parts onto the blank canvas. Order DNA parts such that the 5' and 3' overhangs for each part match. Place part types in the intended final order of DNA parts. Click Assemble on the bottom right of the page. Note that the software tool will only generate valid buildable assemblies based on the 4 base pair overhangs that flank each part upon digestion with the BSA1 enzyme. Navigate to the Plans tab and download the files generated by the tool.